My dear student friends, in today's lecture, let us study the fungal diseases of silkworms. Fungal diseases of the silkworm Bombyx mori caused by the infection of pathogenic fungi are called muscardines or mycoses. The fungal diseases of the silkworms are one of the major diseases threatening cocoon production. The muscardine in silkworms are of different kinds and are identified by the color and appearance of the dead silkworms. The diseases are caused by fungus belonging to different genus and species. The white muscardine, green muscardine and yellow muscardine or aspergillosis are common during rainy and winter seasons in almost all sericultural countries. This disease has wiped out the entire sericulture industry in Italy and France during 1920-25 period. The loss due to the muscardines ranges from 30 to 40% of the total loss due to diseases. Prevalence of 1 to 100% muscardine incidence was observed among the farmers of Karnataka. Further, white muscardine caused by Beauveria bassiana is more prevalent. During 1974-75, due to white muscardine, the sericulture industry suffered a severe loss in Karnataka, but the disease was brought under control by the application of formalin chaff, which is a mixture of formalin and partly burnt paddy husk in the ratio of 1:10 by volume. There is a heavy crop loss during every rainy and winter seasons due to fungal diseases. The mycoses are more when the temperature is low and the humidity is high, which are congenial for their development. For better understanding of the topic, it is being explained in different subheads and let us study them one by one. 1. Infection process. 2. White muscardine. 3. Green muscardine. 4. Aspergillosis. 5. Other muscardines. 6. Alternate hosts. 7. Fungal disease management. Infection process. The mode of infection is per-integumental or percutaneous. Infection by the fungus normally occurs by penetration of the host integument by germ tube from an infective conidium. Pathogen invades the body of the host by producing suitable enzymes and substances exuded by the germ tube. Germ tube secrete protease, lipase and chitinase. The activity of these enzymes are closely related to the pathogenicity to the silkworm. This dissolves the integument and enters into the body. These enzymes or substances are toxic to the silkworm larvae. The pathogen spore germinates by forming a germ tube and penetrates through the integument of the host. Once the pathogen has penetrated the cuticle, it proliferates rapidly in the hypodermal region and grows into mycelium. It invades the hemocoel, destroys the hemocytes and fat body cells prior to death. The internal tissues are then invaded by the pathogen, either growing as a continuous mycelium or as free cells. The free cells may again grow into mycelia. The proportion of free cells and mycelia in the hemolymph is indicative of the stage of the disease. White muscardine. White muscardine in silkworm is commonly known as Sunnakadi Roga in Kannada and Chenaketu or Chitti in Bengali. White muscardine was the first disease described in insects as caused by a microorganism. Agostino Bassi in 1835 demonstrated the contagious nature of the disease. He identified the cause as fungus and suggested measures to control the disease. White muscardine in silkworms is caused by Beauveria bassiana and Beauveria brongniarti that attack all the stages of silkworms. The white muscardine belongs to kingdom fungi, division Eumycota, subdivision Deuteromycotina, class Hyphomycetes, order Moniliales, family Moniliaceae, genus Beauveria, species Bassiana or Brongniarti. Beauveria bassiana produces conidia that are resistant to environmental extremes and are infective stage of the fungal life cycle. The colony of Beauveria bassiana is velvety white, which later turns into yellowish white. The colony of Beauveria brongniarti is velvety to powdery, developing into colony of 10 to 16 mm diameter and a height of about 5 mm. At a temperature of 20 degrees centigrade and 95% relative humidity condition, the pathogen develops very quickly in the host organism. The sources of infection include dead silkworms, contaminated mulberry leaves, infected insects, rearing personnel, rearing equipments and furniture. As a severe contagious disease, it spreads very rapidly among silkworms in a colony, in a rearing room, and from one rearing room to the other or place to places by contact and by air. Contamination of egg surface with conidia results in the infection of just hatched out silkworms. Under favorable conditions, the spores stuck to the cuticle sold in 24 to 48 hours and give out one or two microscopic slender and thin walled germ tubes. These tubes force their way into the body in about 24 hours through spiracles or integument by mechanical force or enzymatic action or both. At the time of invasion, hypodermal cells surrounding the infective hyphae are destroyed. Black oily spots develop due to the drastic disturbance in phenol oxidase activity of the integument. Hypodermal histolysis occurs, resulting in death of the host. Inhibition of circulation resulting from aggregation of conidia in hemocytes and toxins produced are the cause of death of worms. Symptoms: the disease affects all stages of life cycle of silkworm and symptoms appear at the late stage of infection. Larva: as the disease progresses, the spots resembling oily specks are observed on the body surface. The infected larvae lose appetite, become inactive, and on death flaccid. Vomiting and diarrhea also may be observed. Black spots are seen on the body surface at the point of infection. The dead larvae gradually become harder and finally hard and mummifies. The entire body except the head becomes covered with white powdery conidia and the larvae look like a dry white stick. Pupa: the infected pupa loses its sensitivity. The thoracic region shrinks and the abdomen becomes wrinkled. The pupa may develop black spots on its surface and after death it becomes shrunken and the fungus is seen coming out through intersegmental region as pupa mummifies. The mummified pupa is hard, light and white. Moth: the infected moths are sluggish with deformed wings. The dead moth becomes hard, light, mummified and white. Egg: infected eggs are paler and conidia may be observed on the egg surface. The fungal mycelium can be observed inside and outside the diseased egg. The diseased eggs do not hatch. Green muscardine: the green muscardine disease in silkworms is caused by either Metarhizium anisopliae or by Pneumonia raleigh. The colony of Metarhizium anisopliae are dark green, fluffy, cottony and thick. Aerial mycelium is white and fluffy and as it develops with the appearance of fruiting bodies attains dark green in about 10 to 12 days. The green muscardine caused by Pneumonia raleigh belongs to kingdom Fungi, division Eumycota, subdivision Deuteromycotina, class Hyphomycetes, order Moniliales, family Moniliaceae, genus Pneumonia, species raleigh. Under humid temperature of 22 to 23 degrees centigrade conditions, the conidia of Pneumonia raleigh swell in about 8 to 10 hours and germinate by 15 to 20 hours by projecting tubular gem tube. The penetration of the gem tube and its propagation in the host is much similar to Beauveria bassiana. At the early stage of infection, there are no easily identifiable morphological symptoms. As the disease progresses, black spots are developed on the host body. This is due to the lysis due to enzymatic action. Pneumonia relay secretes chitinase, protease and lipase enzymes. By the fourth day, laterally branched hyphae penetrate the endocuticle. The penetration of the gem tube into hemocoel is observed after 5 days of infection. Hyphal bodies are formed by budding from pre-existing hyphae. The hyphal bodies are short, thick and 1 to 3 celled filaments and distinctly nucleated. The blood cells are the first to be invaded, followed by the fat lobes, Malpighian tubules, muscles and mesenteron. The infected larvae lose appetite, become inactive and on death flaccid. Vomiting and diarrhea may also be observed. At the death of the host, hyphae begin to grow outward. The mummified larva appears green, hence green muscardine. The entire body except the head will be covered with the green powdery material and the larvae look like dry green stick. The fungus infects primarily the third and fourth instar silkworms. In case of other stages of infection, the symptoms are similar to that caused by Beauveria bassiana, except that this gives a green color to the mummified pupa and moths. Aspergillosis. Aspergillosis is a mycosis in insects caused by Aspergillus species of fungi. Aspergillus flavus, Aspergillus tamari and Aspergillus ochraceus are some of the Aspergillus species infecting young age silkworms. Aspergillus flavus infecting silkworm eggs is very common. The disease prevails in India is in low intensity. It prevails during winter and rainy seasons in all sericultural areas. The aspergillosis caused by common Aspergillus species belong to kingdom Fungi, division Eumycota, subdivision Deuteromycotina, class Hyphomycetes, order Moniliales, family Moniliaceae, genus Aspergillus, species Flavus or Tamari. Symptoms: Aspergillus species of fungus generally infects early instar silkworm larvae and less common in later instars. Since aspergillosis is caused by various species of Aspergillus, the symptoms are not always same. The diseased early instar silkworms become inactive and lustrous and die without clear morphological symptoms. The body becomes hard at and around the site of infection. It is common on young silkworms because the high temperature and high relative humidity conditions required for the silkworm growth is also quite favorable for growth and multiplication of Aspergillus fungi. As the silkworm feces and litter offer a suitable substratum for growth of this fungus, management of pathogen requires utmost attention. Clusters of mycelial growth is observed after death of larvae and the color depends on the species of Aspergillus involved. The worms die due to the aflatoxin produced by the fungus in the host. The infected first and second instar larvae succumb to death and adhere to the mulberry leaves. Mycelial cluster develops quickly on dead worms. The color of the conidia varies with the species of fungi involved. In the case of third, fourth and fifth instars, which are rarely infected, very characteristic symptoms appear 1 or 2 days prior to death. Infected larvae become sluggish, feed less and localized and hardened areas are seen on the larval body. Hardened area is confined to the spot of invasion, usually either in the middle or posterior end or near the head region of the larva. The corpses become shrunken, hard and result in mycelial growth after 24 hours of death. The growth is initially white, then turns yellow to light brown after 48 hours and to brownish after 72 hours in case of Aspergillus thameri infection. Aspergillus flavus produces green colored conidial spores. The entire cadaver of first and second instar gets covered with the fungus, while in other instars the growth starts from the hardened area and continues to spread on either side to cover the whole body. The pupae rarely get infected, become hard, slightly shrunken with the mycelial growth in the intersegmental and head region. Other muscadines. Apart from the above fungal diseases, some other fungal infections also are observed in silkworms. These minor fungal infections cause varying degrees of damage to the silkworm crops. As the pathogenic fungi involved are different, the disease manifestation, epizootiology and etiology also differs. Let us see white muscadine. The causative fungus is Bacillomyces farinosus. The conidia attaches to the host body. Germ tubes are formed which penetrates into the host. Later mycelia ramify in the internal system. Incubation period is 5 to 7 days in young larvae and 5 to 10 days in grown-up larvae. Late instar larvae are highly susceptible. The toxin produced in the host causes peristalsis in the gut. As the disease progresses, the silkworms lose appetite, become inactive and form small black specks on the skin. Other symptoms are similar to white and green muscadine except that the cadaver is covered with yellowish mycelia and conidia. Red muscadine. The causative fungus is Sporosporella uvula. This pathogen is unique in completing its entire development within the body of its host, producing no growth externally. The body of the infected larvae occasionally presents red patches in a few hours before death. When the insect is broken open, brick red powdery mass of spores escape. The spores germinate upon exposure to moist air. Orange muscadine. The causative fungus is Sterigmatocystis japonica. The infection is usually prevalent in Japan. These invade into bamboo and wood, thus acting as a source of contamination. Early instar silkworms are more susceptible compared to later instar worms. Infected larvae become compact and lustrous and die soon. Body of the infected worms become hard at and around the site of infection. Hirsutella muscadines. The causative fungus Hirsutella saturninensis characteristically produces cinnata which bears lateral phyllites. Conidia are hyaline, single-celled, oblong to cylindrical and covered with mucus. The fungus is pathogenic to early instar silkworms. Black spots appear on the integument of the infected larvae just after death. Long cinemata are produced from the dead larvae. Alternate host. The pathogenic fungi widely infect the wild insects and those infected insects produce great amount of conidia. This results in the persistence of pathogen at a higher rate in the environment. The alternate hosts of fungal pathogens have a great role in developing an epizootic in silkworm rearings during the favorable conditions. It is reported that the natural population of Plusia species, Spilactia obliqua, Diphenia pulverentalis and Echorista bombycis are host to fungal pathogens. These are a major source of contamination of fungal pathogens in the silkworms. The conidia of the pathogenic fungus in the wild could be brought into the rearing room by way of mulberry leaves and winds. Under congenial environmental conditions, they could infect the silkworms at an alarming proportion. Beauveria bassiana insects of 6 orders and causes death to more than 190 insects. Similarly, Aspergillus species infects many agricultural insect species and which is available throughout the year. Further, these pathogenic fungi are saprophytic in nature and grows in all substrata conducive its growth. This also enhances the spore load in the environment, which can easily break into the rearing environment and infect the silkworms, inflicting severe damage. Management of fungal diseases in silkworms. By adopting suitable prophylactic measures, the fungal diseases can be managed and the damage caused due to this infection could be minimized. Though the disease is highly contagious, it has been established that by adopting various control strategies, the disease could be managed very well. High humidity and a high temperature predisposes the fungal infections during young instar silkworms, but this condition is well suited for larval growth. A composite practical technology for silkworm disease management at farmers level was suggested for its efficacy and adaptability. The technology consists of disinfection of rearing house and appliances, early disease diagnosis, personal and rearing hygiene, and use of silkworm body and rearing seed disinfectants. Early detection of pathogens. It is important to detect the pathogen in the silkworm rearing room and mulberry field ecosystem. This will help in devising effective measures which enable the successful prevention of the infectious pathogens. Observing early symptoms of the disease in the colony will aid in removing the infected larvae. This will help in avoiding episodic conditions of the disease. Proper control of bed humidity and temperature. As it is known that the temperature and bed humidity play a crucial role in the manifestation of fungal diseases, it is very essential to manage them suitably to avoid fungal growth. High humidity and low temperature predisposes the fungal infection from third instar onwards. Therefore, it is essential to maintain suitable temperature and humidity conditions to ward off the fungal infection. Application of slaked lime powder in the rearing bed and surroundings can bring down the humidity. Similarly, the mulberry leaf should not be stored inside the rearing room. Use of heaters or heating devices can enhance the temperature in the rearing rooms. Disinfection of rearing room and appliances. The primary source of contamination is the rearing environment itself. Therefore, it is very essential to conduct thorough disinfection of the rearing house and appliances used for silkworm rearing. The pathogen load in the rearing environment could be minimized by taking up disinfection prior to the start of silkworm rearing. Use of chemicals such as calcium hypochlorite, that is bleaching powder solution, chlorine dioxide, calcium chloride, benzalkonium chloride, thosil chloride sodium, sodium hypochlorite, dichloroisocyanuric acid, trichloroisocyanuric acid, etc. could kill the conidia infecting silkworms. Use of bed disinfectants. Treating the larvae with the various chemicals have been attempted to control or suppress the fungal diseases. Rearing seed and silkworm body disinfectants are very effective in controlling spread of diseases in a silkworm colony. Use of sulfur, formalin chaff, lime, etc. were recommended as control measures and preventive measures during the muscardine epidemic of Karnataka during 1975. Dithan M45, Captain, Bavistin, Captaf are some fungicides mixed with slaked lime powder or kaolin at certain proportion and dusted on silkworms after every molt. Different types of bed disinfectants are being recommended by different institutions. Many bed disinfectants such as Resham Kid Aushad, Vijeta Supplement, Ankush, Sanjeevani, Shakti, Resham Jyoti, Labex, Sericillin, etc. are recommended by different institutions. Slaked lime powder is an excellent material which can reduce the bed humidity drastically. Personal and rearing hygiene. In order to limit the damage due to the infection of fungal diseases, it is essential to control the entry of fungal pathogens into the silkworm rearing room. Therefore, personal hygiene is very important. Utmost care is required to avoid aspergillosis infection during young instar silkworm rearing. Thorough washing of hands before and after entering the rearing house, picking up and proper disposal of infected worms are important. Controlling alternate hosts. Alternate hosts of the fungal pathogens are an important factor in occurrence of muscardine diseases. Further, the use of rearing seat and silkworm body disinfectants can prevent the spread of fungal diseases in a silkworm colony. Thus, the topic can be summarized that muscardine disease in silkworms are caused due to the infection of fungal pathogens. It inflicts severe damage to the silkworm crops. Generally, low temperature and high humidity in the rearing environment are the predisposing factors for fungal diseases. Vivaria bassiana causes white muscardine and Pneumonia raleigh causes green muscardine in silkworms. Different Aspergillus species such as Aspergillus flavus, Aspergillus tamari, etc. attack the young instar silkworm larvae to cause aspergillosis. Apart from this, many other fungal pathogens are also causing various fungal diseases in silkworms. There are many management strategies to combat this problem. Reduction of bed humidity, disinfection of the rearing environment, use of bed disinfectants are some of the management strategies. Thank you.